All right, I'm going to go ahead and get started. Before I introduce Lauren, I just would like to remind everyone about some of the Zoom seminar etiquette. So all of you guys will be muted throughout the seminar, so please do not attempt to turn on your video or share your screen during the talk. There will be an opportunity for you to ask questions live at the end of the talk, just like in a normal seminar. So once the speaker has finished their talk, you can use Zoom's raise hand feature to notify the meeting host that you'd like to ask a question. And this raise hand feature is located under the reactions tab at the bottom of the Zoom window. And you are invited to turn on your video during the questions portion of the talk. So now getting to the fun part, it is my pleasure to introduce Lauren Cooley today. I'm excited for you guys to hear about the research that she's been doing over the last few years. So I first met Lauren a few years ago when she responded to an announcement I had posted where I was looking for a graduate school student to work on our diving physiology project of working with elephant seals. She had a bachelor's from Cornell in animal science, providing her with a really strong foundation of physiology, which made her ideal for this position. And then on top of that, she had 2 years of experience working with stranding programs in Florida and Mississippi that kind of provided her with a skill set that would also be very valuable for the Vertebrate Ecology Lab stranding program. So based on this, she was a perfect fit for the program and the Vertebrate Ecology Lab. So I was very excited when she accepted the offer to join our lab. As many of you guys know, Lauren is very organized and detailed oriented, which is something that I very much appreciate and can use help with. So she quickly joined, kind of whipped our lab into shape and helped get me into shape. She did bring her stranding network expertise, and she worked closely with Sean and later Daphne to really get our stranding program kind of up with current protocols and making sure we were collecting the best data and entering it in a timely fashion. So Lauren was often out in the field or helping with necropsies and managing this data. And she trained kind of most of the incoming volunteers and has really kind of helped grow this program. Because of her detailed orientedness, I also encouraged her to become the lab safety manager. So since joining the lab, she really helped clean up the lab, although if you looked at now, you can see that she's gone. It's gone downhill since she's left. But the reason she came to the lab was to join us on our elephant seal project. And she joined graduate student Sean and became the eSeal Power Duo. I think she was maybe a little intimidated that first year when she had to go through and they went through and inventoried all our surgical supplies and they didn't know what half of it was, but did a great job. This duo eventually became a trio when Daphne joined the project. And Lauren really put her lab safety skills and management into practice. Anytime they had to construct new equipment, they made sure it could handle the weight. And I loved getting little kind of texts with photos like this, keeping me updated on their progress. She is also a lab tech extraordinaire. She processed hundreds of samples for her own thesis and also for the collaborators on this project. She is an excellent, her lab safety manager skills definitely were applied during this field project. Here you can see her demonstrating all of the safety equipment that you need when you're dealing with liquid nitrogen. And best of all is on this project, she was very excited about her samples. So in the picture on the left, you can see she's getting hematocrit measurements. And when the labs finally started to open up and she could get her samples, she was very excited about all the samples she had collected. In addition to working on our elephant seal project, she helped out with a lot of different projects. So here's a photo of one of the wiener weighing teams. And sometimes she even offered up to lead morning exercises to make sure no one would get hurt in the field. So you can see her demonstrating, warming up your arms. In addition to being hardworking, she also did make sure to participate in lab events and have fun and relax, especially during COVID. A lot of this involved getting outdoors. And during that time, she made a lot of good friends. And because we don't necessarily have the typical opportunity to do the slideshow, I thought I'd throw in some of these here. She will be a missed member of the Vertebrate Ecology Lab. She's been part of our 3 awkward family Christmas photos, and we'll miss her in our next one. And then just to sum up some of her accomplishments and awards while at Moss Landing, in addition to being an excellent scientist, she also is a good community member. So she has coordinated blood drives, she's managed our stranding network, she received a Coast Graduate Student Award, a Moss Landing Wave Award, a Simpkins Family Marine Science Award, Myers Oceanographic and Marine Biology Trust, and just recently, when presenting her work at the Experimental Biology Conference, she received a Comparative and Evolutionary Physiology Section Research Recognition Award. And she's already moved on and left us. She is working as a stranding biologist with IFAW on the East Coast. So she is helping to kind of establish this Bell satellite group now that is establishing itself in Cape Cod. And with that, I am excited for Lauren to kind of take over and share what she's been doing for the last few years.
All right, well, thank you so much, Gita, for that very warm introduction. Give me a second here while I go ahead and share my screen and get situated. All right, just one sec here. And I think we are almost good to go. Okay, so thank you all so much for gathering here today. I'm really excited that you've all joined me for the defense of my master's thesis titled Research Handling Effects on Stress Hormones, Blood Parameters, and Heart Rate in Juvenile Northern Elephant Seals. So let's get started. We'll begin now with a short introduction. In an ideal world, wildlife researchers would be able to collect all the data they need from their study animals without causing any form of disturbance. While researchers collecting certain types of non-invasive behavioral or acoustic data, or even certain non-invasive samples such as feces, may be able to meet this gold standard, the reality is that the majority of physiological and behavioral studies of wild animals require some degree of animal handling to collect blood or tissue samples, measure animals, or to attach instruments such as satellite tags or physiological biologgers. And throughout this talk, I will be referring to those various techniques as research handling. Marine mammal research in particular often requires a high degree of research handling, as instrumentation provides one of the only means of collecting crucial behavioral and physiological data from at-sea animals. When we think about research handling, it's really important to consider the impacts of these activities on our study animals. So while capture and handling are essential components of marine mammal research that advance both science and conservation initiatives, these activities can be stressful for study animals and have the potential to alter both behavior and physiology, and in some instances may cause injury or death. So when I say stressful, what exactly does that mean? It's actually harder than you might think to define stress, but for the purposes of this talk, we will be defining the stress response as the suite of physiological processes and behavioral responses elicited by an organism when confronted with a challenge to homeostasis called a stressor. When we talk about stress, it's really important to consider the time scale that these responses are occurring on and to distinguish between short-term and long-term stress responses. So an acute stress response to a short-term stressor can actually be an adaptive ability and allow that animal to cope with a challenge, and that will help them prioritize individual survival over longer-term objectives, such as energy storage, growth, or reproduction. However, a chronic stress response to a persistent stressor can actually be maladaptive and compromise body condition, immune function, reproductive success, and ultimately survival. So you might be asking, how does the stress response actually work? And a very simplified answer to that question is that the stress response is mediated by hormones. Hormones are signaling molecules used by the body to communicate between organs and tissues. Hormones regulate a number of physiological processes and behavioral responses, including the stress response. And I just wanted to note here that throughout this talk, you may hear the phrase endocrinology, and that simply refers to the study of hormones and the endocrine system, which includes many of the organs and glands shown here in a cat. So I'm going to guess that one element of the stress response that many of you may be familiar with is the so-called fight or flight response. So what exactly does that entail? The fight or flight response is an automatic physiological response to a stressor. This involves a rapid hormonal cascade involving the catecholamine hormones epinephrine and norepinephrine. And the fight or flight response has a number of physiological effects on the body, including increased heart rate, increased respiration rate, and increased blood glucose. In addition to the fight or flight response, another component of the stress response is mediated by the HPA axis. HPA stands for hypothalamic pituitary adrenal axis, so it's a bit of a tongue twister, which is why we say HPA. And the HPA axis controls a complex hormonal cascade that culminates in the release of glucocorticoids, which are steroid hormones produced by the adrenal gland. In mammals, the most common glucocorticoid is cortisol. Cortisol has a number of physiological effects on the body, including increased blood glucose and simultaneously decreasing energetically costly functions such as growth, reproduction, and immune functioning. So when it comes to measuring and quantifying stress responses, researchers use a variety of different approaches that I'll briefly review here. So to begin with, to measure stress, you could look at changes in physiology, such as hormones, heart rate, or blood parameters. You could also look at changes on a molecular or cellular level, such as DNA damage or repair, or antioxidants. On more of a whole animal level, we could look at changes in behavior, such as changing activity patterns, or vocalizations, or stereotyped behaviors. And finally, we could look at changes in fitness, such as injuries, or even changes to larger population demographics, such as birth rate or death rate. But while there are merits to all of these different approaches to measuring stress, today you're in a physiology talk, so we'll be focusing in on hormones, heart rate, and blood parameters as metrics for stress. So to begin with, let's talk a little bit about using hormones to measure stress. Endocrinology is the most common approach in stress research. And one of the major advantages of using hormones to study stress is that they can be measured in a variety of different tissue samples, including blood, saliva, and fur. And depending on the sample type that you use, hormones can be used to study stress across varying timescales, including both acute and chronic stress responses. And in this project, I measured two different stress hormones, cortisol and aldosterone. Another approach used by researchers to measure stress is looking at changes in blood parameters. Studying blood parameters allows researchers to understand the downstream effects of stress on physiological processes, such as metabolism, immune function, inflammation, and oxygen transport. In this study, I measured 5 different blood parameters that can be broadly grouped into 2 categories. In the blood chemistry category, I measured blood glucose, ketone bodies, and blood lactate. And in the hematology category, I measured hematocrit and erythrocyte sedimentation rate, or ESR. And one of the final physiological metrics that we'll discuss today that we can use to quantify stress levels is heart rate. Elevated heart rate, or tachycardia, is an immediate indicator of stress. So if you've ever been startled by something and felt a spike in your heart rate, you know exactly what it means to experience tachycardia. And unlike hormones and blood parameters, which tend to be measured periodically, heart rate data can be measured continuously. And these continuous measurements allow for more rapid and fine-scale analysis of the effects of specific short-term stimuli on stress levels. So in this project, I calculated a variety of heart rate parameters. For my longer-term stage analysis, I calculated mean heart rate and interbeat interval range. And for my short-term stimulus analysis, I calculated mean heart rate. So now that we reviewed some basic elements of stress biology, let's focus in on how stress relates to the research handling activities we discussed earlier. So if you were to review the existing stress literature or simply ask the average wildlife biologist, you would find that capture and handling activities are well-known stressors for wild animals. Even small degrees of handling stress have the potential to introduce physiological stress artifacts, which are unintended changes in the physiological parameter caused by the researcher or the experiment rather than nature. And from a research perspective, it's really important here to acknowledge that stress artifacts have the potential to both bias and confound research results. So for example, say you are a researcher studying cortisol levels in a population of wild bottlenose dolphins, such as the animal shown here. To do this, you could measure cortisol levels in blood samples collected from a subgroup of animals in this population. However, if the act of capturing those dolphins and collecting the blood samples increase the stress levels of these animals, your measured cortisol values are likely not going to reflect true baseline. And that's just one example of how these physiological stress artifacts can bias research results. So, why is it important to study handling stress? I would contend that researchers have an obligation to both examine the impacts of their research activities on study animals and to continually refine their protocols to reduce stress whenever possible. And there's a number of benefits that come with reducing handling stress. So first of all, you're going to improve your science. By minimizing any confounding effects of physiological stress artifacts, you're going to have a stronger, more robust study. Reducing handling stress will also help maximize animal welfare for all of the individual animals involved in your study. And on a broader scale, by improving the welfare of individual animals, you can help further conservation goals on a population or even species level. So all of that brings me now to my study. Upon reviewing the existing literature, I found that the effects of research handling activities on marine mammals were rarely analyzed as a complex physiological interaction. In response to this gap in the literature, I developed my study using a holistic approach. My goal was to integrate a diverse set of physiological parameters traditionally analyzed separately into a single comprehensive assessment of the acute stress response to handling in a model marine mammal species. And that model species was my beloved northern elephant seal, Mirounga angustirostris. So I'd love to tell you a little bit more about these amazing pinnipeds. Northern elephant seals are phocids, or true seals, and they are the second largest pinniped species in the world, second only to their cousins in the southern hemisphere, the southern elephant seal. Elephant seals exhibit extreme sexual dimorphism, in which the males are substantially larger than the females. Male northern elephant seals can weigh up to 2,300 kgs, which is about 5,000 pounds. So they're just truly massive animals. Northern elephant seals range across the North Pacific from Alaska to Baja California. And they have really interesting annual cycles in which the seals spend the majority of their time at sea, but they do come to shore twice per year to breed and molt. And when they're on shore, they tend to aggregate in large colonies or rookeries. Northern elephant seals are extremely well studied and are considered by many researchers to be a model species for ecological and physiological research. So what makes the northern elephant seal a model species? To begin with, they are readily accessible to researchers when hauled out on shore in their large colonies. They also tolerate instrumentation well, and their predictable annual cycles facilitate the recovery of instruments by researchers. And finally, elephant seals have documented cortisol responses, which is particularly relevant for stress physiology projects like mine. At the elephant seal colony at Año Nuevo State Park, over 500 northern elephant seals are handled per year. Research handling techniques used by scientists at Año Nuevo include chemical immobilization, physical restraint, instrumentation with biologgers, and translocation studies. So now that we've reviewed some basic principles of stress physiology, marine mammal research handling, and elephant seal research in particular, I'd love to quickly review the 4 objectives of my project. First, I wanted to quantify hormonal stress responses using periodic cortisol and aldosterone measurements. Then I wanted to measure cardiac stress responses using continuous electrocardiogram or ECG heart rate data. Then I wanted to examine the effects of stress on whole body physiological processes using blood parameters. And finally, I wanted to determine the relationship between cortisol and the other study parameters such as aldosterone, heart rate, and those 5 blood parameters. So with that being said, let's get into the methods and discuss how exactly I did this. So my study was conducted as part of a much larger project that was funded by the National Science Foundation. The focus of this larger project was using at-sea experimental disturbances to characterize physiological plasticity in diving northern elephant seals. And before we go any further into the methods, I did want to briefly note here that marine mammals are federally protected by the Marine Mammal Protection Act, and that all of the research activities that you'll be hearing about in this talk were permitted by the National Marine Fisheries Service. So with that being said, let's get into a few quick stats about this project. This study was conducted over 2 field seasons in 2019 and 2021. We had a total sample size of 19 juvenile northern elephant seals, of which we had 4 males and 15 females. And a little bit more about our seals. On average, they were 1 year of age, and they weighed on average 170 kgs or 374 pounds, and measured on average 185 centimeters straight length or 6 feet. So while they were juvenile animals, they were still quite large. This study utilized a well-established translocation protocol. So I'm going to quickly review the steps in a translocation, and then we will go through each step in more detail. So all our translocations began at Año Nuevo, where we went up and captured a seal via chemical immobilization. The seals were then transported to Long Marine Lab at UC Santa Cruz, where they underwent a 3-hour instrumentation procedure to attach a series of biologgers. After the procedure, seals were covered in their cage and then were transported to Pacific Grove, where they were released. After the release, the seals swam across Monterey Bay and returned to the colony at Año Nuevo. And then once they returned to the beach, they were recaptured a final time, and we recovered our instruments in the field. So now that we've quickly reviewed that whole translocation protocol, let's go through each step in a little bit more detail. So the start of every translocation began with capture at Año Nuevo State Park. To do this, researchers from our team would go out and select a healthy, unmolted juvenile seal, 1 to 2 years of age, On the beach, we would chemically immobilize the seal with intramuscular tilazol, and once sedated, the seal would be rolled into a secure metal transport cage. Next, we would transport the seal to the lab. So we did that via truck. We would load the cage onto the truck, and then the truck would drive around 30 kilometers to Long Marine Lab in Santa Cruz at UC Santa Cruz's Coastal Science Campus. Following arrival at the lab, the seal would be unloaded and weighed in the cage, and then we would move the cage into a quiet animal holding area and allow the seal to rest for several hours. Following that short holding period, we would begin our instrumentation procedures. These procedures are conducted under full anesthesia. We would start out by initially immobilizing our seals with intramuscular tilazol, and then from there, they would be induced with a mask, which is shown here in the photo, and then intubated with 5% isoflurane oxygen gas anesthesia. These instrumentation procedures were pretty long, lasting on average 3 hours with a range of 2.5 to 4.5 hours. During the procedures, we would collect measurements and attach a series of tags to our seals. Once the seals were anesthetized, we would collect a few basic morphometric measurements, including a straight length and a series of girths. We would then apply flipper tags with unique identification numbers to the hind flippers of any animals that had not been previously tagged as part of another study. And then the vast majority of those procedures were spent attaching 5 different biologging instruments. We would attach those to the fur using a marine epoxy. So I'm now going to briefly review those 5 instruments that we would attach during each procedure. On the head, we would attach a satellite tag that would be used to locate the seals as they swam across Monterey Bay. Next, we would attach a time depth recorder, or TDR, which would collect dive behavior data from our animals as they moved the water column. Near the TDR, we would attach an AMX acoustic tag, which collected acoustic and acceleration data, and also played an acoustic disturbance sound while the seals were diving. Behind that, we would attach a very high frequency, or VHF, radio transmitter, which was used to locate our seals on the beach once they returned to Año Nuevo. Finally, the most complex instrument we would use was the Oxylog physiological logger. This instrument was connected to 3 electrodes that measured the electrocardiogram heart rate, venous oxygen, temperature, and ambient pressure. And then at the end of each procedure, the 3 electrodes would be secured and covered with a neoprene patch. And so that is what all of our seals would look like at the end of their instrumentation procedures. The next step in the translocation is the post-procedure recovery period. So at the end of anesthesia, the seal would be moved back into the cage and then manually ventilated with 100% oxygen until the recovery of spontaneous breathing, at which point they would be extubated. Next up would be the recovery. And so after the procedures, our seals would recover under observation for a minimum of 4.5 hours. 12 of our study animals recovered the same day as their procedure, and the remaining 7 animals recovered overnight. Once the recovery period was finished, it was time for transport and release. So for transport, we would load the cage onto the truck, and then the truck would drive around 70 kilometers to a sandy beach release site in Pacific Grove. And then for release, our team would carry the cage down the beach and set it down near the surf. And then once the door was open, our seals would pop out and head down into the water. So I really enjoyed the entire translocation project, but one of my favorite moments were definitely our releases, which typically occurred at either sunrise or sunset. So while I take a brief sip of water, I'm going to show you a short clip from one of our sunrise elephant seal releases. So here you can see our seal is coming out of the cage. He has all of his instruments on him, and he is heading back into the water, where he'll shortly begin to swim northward back to the elephant seal colony at Año Nuevo, while all those tags that are attached to him collect a bunch of valuable physiological data for us. So moving on to the final step of every translocation, which is the recapture. So for the return to Año Nuevo, our seals would swim across Monterey Bay, and that swim would typically take them anywhere from 2 to 10 days. And while they were swimming, the biologgers attached to them would be collecting data during a series of continuous dives. Once the seals returned to Año Nuevo, it was time for the recapture. So we would locate the seal on the beach using the VHF transmitter. And then finally, we would immobilize them one more time with intramuscular tilazol and recover those instruments in the field. And while our translocations did officially end with the recovery of our instruments, luckily, we were able to collect additional follow-up data on our animals post-translocation. So post-translocation study animals were opportunistically resited in the field through daily colony survey efforts led by UC Santa Cruz researchers, and individual seals were identified using their unique flipper tags that we talked about earlier, which you can see in the top right corner. So now let's discuss some of the samples that we collected throughout these translocation procedures. Throughout each translocation, periodic blood samples were collected from the extradural vein at up to 6 time points. I'm going to quickly go through those 6 time points now. The first blood sample was collected in the field at capture. Our goal was to collect the sample as quickly as possible following initial disturbance. And because we were able to collect those samples so quickly, this sample actually served as the baseline level that we used to compare later samples to. Our next two samples were collected during the instrumentation procedure. The early procedure sample was collected 50 minutes after gas anesthesia started, and the late procedure sample was collected 2.5 hours after gas anesthesia started. The next two samples were collected during the post-procedure recovery period. The early recovery sample was collected 3 hours after the isoflurane gas was turned off, and the late recovery sample was collected early the next morning from that subset of animals that were held overnight for recovery. And finally, the last blood sample was collected during recapture. Like the capture sample, this sample was collected in the field as quickly as possible following the initial disturbance. And I did briefly want to note here that those capture, procedure, and recapture samples were all collected directly from the extradural vein using a spinal needle, while the two recovery samples were collected using an extradural catheter and extension set that were left on the animals while they recovered in their cages. So the next big step in my research project was conducting the hormone analysis. This began with a bit of initial blood processing. So we would centrifuge the vacutainers that the blood samples were collected into. And then from there, we would aliquot the serum into cryovials. Those cryovials were stored at -80°C until analysis. And then from there, I performed a series of radioimmunoassays, or RIAs, to measure serum cortisol and aldosterone concentrations. I performed these assays in Dr. Dan Crocker's lab at Sonoma State University, and all samples were analyzed in duplicate. In addition to the hormone analysis, I also used the blood samples to measure a series of 3 blood chemistry parameters. Blood glucose and beta hydroxybutyrate blood ketones were measured using a Precision Extra monitoring system, and blood lactate was measured using a lactate plus meter. I also measured 2 hematological parameters using those blood samples. Erythrocyte sedimentation rate, or ESR, was measured using a steady rate Westergren ESR system, and hematocrit was measured using microcentrifugation. In addition to the hormones and blood parameters, the other main focus of my project was looking at stress effects on heart rate. So the OxyLog custom physiological logger manufactured by UFI Instruments was used to collect the electrocardiogram data, and that data was collected continuously at a frequency of 100 Hertz. And once the data was collected, it was processed and converted into instantaneous heart rate in beats per minute using custom scripts in MATLAB and R. When it came time to analyze my heart rate data, I broke down the analysis into 2 main areas: a longer-term stage analysis and a shorter-term stimulus analysis. The focus of the longer-term stage analysis was to characterize general heart rate trends during different stages of the experiment. And to do this, I calculated 2 variables, mean heart rate and interbeat interval, or IBI range. And I calculated those parameters during 5 different stages of the experiment: late procedure, and then 3 different stages of the recovery: early recovery, mid-recovery, and late recovery, and finally transport to the release site. In addition to that longer-term stage analysis, I also performed a short-term stimulus analysis. And the focus of this was to examine the immediate effects of 7 short-term stimuli on cardiac stress levels. And to do this, I calculated 2 different variables. I calculated mean heart rate during the stimulus event and mean heart rate during a 5-minute baseline period directly before the event. I wanted to note here that while I do use the term baseline, this doesn't actually refer to baseline heart rate levels as would be seen in wild animals. This is simply baseline relative to any additional stress from those short-term stimuli. The 7 short-term stimuli that I examined were extubation, or the removal of the tube at the end of anesthesia, the collection of our blood samples during the recovery period, when we would turn on the light at the end of overnight holding, and then 3 different stimuli during the transport process. So loading the cage onto the truck, the first few minutes driving the truck, and then the midway stop that we would make on our transport. And the final stimulus I looked at was the release. And finally, to wrap up the methods, I wanted to very briefly touch on the stats that I used. So all statistical analyses were performed in R, and statistical significance was accepted at an alpha level of 0.05. And I want to very quickly review the specific statistical tests that I used. So first of all, I used linear mixed models and Tukey post-hoc tests to analyze changes in hormones, blood parameters, and heart rate over the course of the translocation among repeated samples. Next, I used paired t-tests to analyze my heart rate stimuli data. And finally, I used mixed model linear regressions to analyze the relationship between cortisol and the other study parameters. So that now brings us to the results. So what exactly did I find? To begin with, I want to very quickly review how to read a box plot, as you'll be seeing an awful lot of box plots in this results section. So on the x-axis of all our box plots, we have the experiment stage, which includes those 6 time points that we talked about earlier with the blood samples. On the y-axis, we have our study parameter of interest, and then we have a series of box plots. So you'll see each time point has its own box plot. That center bold line is the median. The colored box includes the interquartile range, which is the 25th to 75th percentile. And I want to briefly note here that the wider or taller the box is, the more variation. So we can see here, this dark green box has a very high degree of variation. And then on our boxes, the outer whiskers show the minimum at the bottom and the maximum at the top. Any dots are outliers. And then finally, the small lowercase letters above each box show group differences. So now that we know how to read a box plot, let's get into my hormone results. So first, we'll discuss cortisol. Cortisol is a steroid hormone secreted by the adrenal glands, and it is considered the primary mammalian stress hormone. Looking at our cortisol results, in general, I found a very wide range of cortisol values from less than 27.6 nanomoles per liter up to over 2,500 nanomoles per liter. So that's a very substantial range. At capture, we see that our baseline cortisol levels were quite low, with an average of 183 nanomoles per liter. Cortisol was significantly elevated above baseline during the 2 procedure samples shown here in blue. Cortisol reached its peak of 806 nanomoles per liter during the early recovery stage shown here in light green. And this was a 4-fold increase over baseline capture levels. And I'd also like to note that the 2 highest individual cortisol levels measured in this study, shown here as the 2 outlier dots at the top of this graph, both occurred during this early recovery stage. However, for the seals that were held overnight for recovery, by the next morning in the late recovery stage, which is shown here in dark green, cortisol had actually returned to baseline level, and at recapture, cortisol levels stayed at this baseline level. So next up, we'll discuss aldosterone. Like cortisol, aldosterone is a steroid hormone secreted by the adrenal glands. While the primary function of aldosterone is water regulation, there's actually increasing evidence that aldosterone is a component of the acute stress response in marine mammals, which is why I chose to include it in this project. I found a pretty wide range of aldosterone values from less than 66 picomoles per liter up to over 1,200 picomoles per liter. At capture, baseline aldosterone levels were quite low at around 193 picomoles, and aldosterone was significantly elevated above baseline during the early procedure stage, and it reached its peak of 604 picomoles during the late procedure stage, which is shown here in dark blue. And that was a 3-fold increase above baseline levels. Aldosterone remained elevated during the early recovery stage, and unlike cortisol, aldosterone did not return to baseline levels during the late recovery. Although you can tell from that very tall dark green box that there was a high degree of variation between individuals at this stage. However, by recapture, aldosterone did return to baseline. Now we'll move on to the blood parameter results. So first up, we'll talk about hematocrit. Hematocrit is the percentage volume of red blood cells in blood. I did want to note here that relative to most other mammals, pinnipeds have extremely high hematocrits, and that hematocrit regulation in pinnipeds is quite complex, but past research has indicated that it may be influenced by both research handling method and by anesthesia, which is why I chose to include it in this study. So here I found that hematocrit values ranged from around 44 to 74%. And in general, when we look at this figure, we can see that relative to those previous plots we looked at, our box plots are roughly at the same level, which indicates there is relatively small variation in hematocrit levels between the translocation stages. And in fact, hematocrit was significantly elevated above its baseline level of 55% during only one stage of the procedure, and that was the early recovery stage, at which point it reached 60%. And at all other stages, including the procedure stages and recapture, hematocrit remained at baseline levels. Next up, we'll discuss erythrocyte sedimentation rate, or ESR. ESR is the rate at which red blood cells and anticoagulated blood descend in a standardized tube over a period of 1 hour. It's a bit complicated, but the main thing that you need to know about ESR is that it is a non-specific measure of inflammation, and that higher ESR indicates higher inflammation. So while I did find a pretty large range of ESR values in this study from 7 to 113 millimeters per hour, with a mean ESR of 39 millimeters per hour, when we looked at all the different stages, I found that their experiment stage had no effect on ESR. So there were no differences between time points for this parameter. Moving on to blood glucose. Glucose is the main blood sugar. We know that one of the primary effects of cortisol is to mobilize energy stores, which leads to an increase in circulating blood glucose. Because of that, I predicted we would see an increase in blood glucose with stress. Looking at our results, I wanted to first point out that we were unfortunately unable to measure any of the 3 blood chemistry parameters at capture or recapture. So because of that, you'll see for glucose and the next 2 parameters that I'm only going to show data from the 2 procedure samples and the 2 recovery samples. But with that being said, blood glucose ranged from 3.5 to 12 millimoles per liter and was generally lower during the procedure stage, shown in blue, and higher during the recovery stages, shown in green. Blood glucose reached its peak during the early recovery stage, at which point it averaged 7.4 nanomoles per liter, up from only 5.5 nanomoles per liter during the early recovery stage. Now we'll talk about blood lactate. Lactate is a byproduct of anaerobic cellular metabolism. It serves as an excellent indicator of extreme muscle exertion or tissue hypoxia. In this study, I found that lactate ranged from 1 to 3.7 millimoles per liter and was higher during the procedure and lower during the recovery. Lactate reached its peak of 2.7 millimoles per liter during the late procedure stage, just shown here in dark blue. And the final blood parameter that I measured was ketone bodies. In general, ketones are metabolites produced by the liver when glucose is not available. And more specifically, beta hydroxybutyrate, or beta HBA, is one of the main ketone bodies in mammals. For our purposes, the main thing that you need to know about ketones is that they're indicators of fat metabolism. Looking here at our results, my main finding was that experiment stage had no effect on ketones. Beyond that, ketone values measured in this study ranged from 0.1 to 0.6 millimoles per liter, and overall mean ketone was 0.3 millimoles. And now we'll discuss some of my heart rate results. So to help guide my heart rate analysis, I first created heart rate profiles depicting instantaneous heart rate versus time for each study animals. I'm going to now show you the heart rate profiles for 2 different seals. I know these plots might be a little bit overwhelming, but I'm going to walk you through them step by step, so don't worry. So here we have heart rate profiles from 2 different seals. Each row is a different seal, while each column is a 1-hour section of heart rate data from 4 different stages of the translocation. On the far left, we have the late procedure, then early recovery, then mid-late recovery, which are 2 stages that I grouped together due to similarity of the data. And then finally, on the far right, we have transport to the release site. In each of these 8 separate plots, on the x-axis, we have the time of day in hours, and on the y-axis, we have heart rate in beats per minute. So now let's discuss some of the general trends that we can see looking at these 4 different stages of the translocation. During the late procedure stage on the far left, heart rate is very low and stable, but then it begins to gradually increase once the isoflurane gas is turned off. During the early recovery stage, the heart rate continues to increase and becomes more variable than it was during the previous stage. During the mid-late recovery stage, we see a really unique and interesting pattern here of periods of increased heart rate or tachycardia, followed by periods of decreased heart rate or bradycardia. These patterns are indicative that the elephant seals are experiencing sleep apneas, which are a very normal physiological occurrence for this species. And as a scientist, it's actually great to see that these seals are likely sleeping during this stage. It shows they're not too terribly stressed out. And finally, when we look at our heart rate data during the transport, we can see that our seals exhibited 2 different heart rate responses. So the seal on the top seemed to have relatively variable heart rate for the beginning portion of the drive, but then after about 20 minutes, the heart rate seemed to stabilize at a relatively low level. In contrast, the seal on the bottom had a much more variable heart rate level throughout the entire transport process, which did not ever seem to fully stabilize. So now that we've looked at those example heart rate profiles from our 2 seals, let's talk a little bit more about some of the general trends from these heart rate stages. So to do this, I calculated mean heart rate by taking the total number of heartbeats in a given stage and simply dividing by the length of that stage. Overall, mean heart rate ranged from 44 to 93 beats per minute. During the late procedure stage, shown in dark blue, mean heart rate was 50 beats per minute, which was significantly lower than any of the later stages, indicating that the anesthesia caused bradycardia. Mean heart rate then increased during the early recovery stage and increased a bit more in the mid-recovery stage, at which point it stabilized at around 73 beats per minute. During transport, shown here in purple, mean heart rate was 77 beats per minute. While this level was not statistically different from mean heart rate during the late recovery stage, shown in dark green, as you can see from that much taller purple box, there was more individual variation during transport, which if you think back to those different variable stress responses that we saw in the heart rate profiles, that kind of makes sense. Now we'll discuss the interbeat interval range. Interbeat interval, or IBI, refers to the time between consecutive heartbeats. So interbeat interval range is similar to all ranges, is simply maximum IBI minus minimum IBI. I know this parameter might be a little bit more confusing than mean heart rate, but the main thing that you need to know is that interbeat interval range is a simple measure of variability, and that lower interbeat interval range means lower variability. So looking at our results, I found that IBI range was significantly depressed during the late procedure stage, shown in dark blue. During this stage, IBI range was only 0.3 seconds, which indicates there was very low heart rate variability during anesthesia. IBI range then increased significantly during the early recovery stage and remained at this level of around 1.8 seconds during the mid and late recovery and the transport stages. And the final heart rate results we're going to discuss here are the results of my short-term stimulus analysis. So for this analysis, I analyzed the effects of 7 short-term stimuli on mean heart rate levels. For each stimulus, I calculated a baseline mean heart rate and an event mean heart rate. The baseline mean heart rate included the 5-minute time period preceding each stimulus, while the event mean heart rate included the length of the stimulus. So as you can see here, our results are depicted on a bar plot. On the x-axis, we have our 7 stimuli, and on the y-axis, we have mean heart rate. For each stimulus, the baseline mean heart rate is shown in the light gray bar on the left, and the event mean heart rate is shown in the darker gray bar on the right. Well, I found that mean heart rate increased very slightly from the baseline period to the event period for all 7 stimuli. This increase was statistically significant for only the release stimulus, which is shown on the far right end of the plot below the dark blue asterisk. And during the release stimulus, mean heart rate increased from around 68 beats per minute during the baseline to around 77 beats per minute during the event period. And now we'll discuss the relationship of some of the other parameters in this study to cortisol. So using mixed model linear regressions, I found that 3 study parameters were positively associated with cortisol. These parameters were aldosterone, hematocrit, and blood glucose. The remaining 4 study parameters were not associated with cortisol. These parameters were ESR, blood lactate, ketone bodies, and mean heart rate. And finally, to wrap up our results, I wanted to very briefly review some of our post-translocation recite data. So looking at our recite data that was collected from our seals after the end of their respective translocations, we found that 18 out of 19, or 95% of our study animals were recited in the wild post-translocation. We focus in on just 2019 and those 11 seals, 10 out of 11 seals were recited the following year, which is just a wonderful statistic. And finally, to end on a very happy note, one of the seals that we translocated in 2019 was actually seen again in 2021 with a pup. All right, we've made it to the discussion section. So let's talk a little bit about what all of these results actually mean. So first we'll talk about my hormone response, or the hormone response. So in response to capture and handling, the seals in this study displayed a moderate hormonal stress response for both cortisol and aldosterone. Cortisol peaked in the early recovery stage at 4 times baseline levels, while aldosterone peaked slightly earlier in the late recovery stage at 3 times baseline levels. And finally, I found that aldosterone was positively associated with cortisol. So what conclusions can we draw from that hormone data? First of all, the seals in this study displayed a moderate hormonal stress response to capture and handling, but most individuals recovered rapidly and displayed no long-term impacts. I also found that isoflurane gas appears to be an excellent anesthetic agent for northern elephant seals and seemed to really help prevent some of the more extreme stress responses that have been seen in the past with other sedation methods commonly used in pinniped research. And in general, the results of my study were well aligned with previous studies of pinniped handling, particularly when it came to aldosterone. So aldosterone was positively associated with cortisol, which supports previous research findings that suggest that aldosterone is a component of the acute stress response in marine mammals. However, I think that a lot more research is definitely necessary in this area to kind of figure out the exact function that aldosterone is playing in the stress response. And finally, thinking back on the study, if I could do it all again, I really wish we would have been able to collect a blood sample after transport to the release site to see how those stress hormones might have varied following that hour-long drive. The main reason we weren't able to collect that sample was simply due to study logistics, but I just found the heart rate data during that transport to be so interesting, and I'm just so curious as to what would have been happening with the stress levels. So now we'll discuss some of the blood parameter responses that I saw. So hematocrit, glucose, and lactate all exhibited mild responses to handling, while ESR and ketones seemed to exhibit no response. Hematocrit and glucose both peaked during the early recovery stage, while lactate peaked during the late procedure stage, and there were no differences between stages for ESR or ketones. And finally, hematocrit and blood glucose were positively associated with cortisol, while there was no association with cortisol for lactate, ESR, or ketones. So what conclusions can we draw from that blood parameter data? So it appeared that in general, seals displayed a very mild physiological response to handling as measured by 5 different blood parameters. This study reported the first ESR values measured for this species, and very interestingly, the lowest and highest ESR values measured in this study were actually from the same animal. So at capture, that seal's ESR was a very low 7 millimeters per hour, while at recapture, it had spiked to 113 millimeters per hour. And looking back on the notes for the animal, I actually found that our team noted that upon recapture, that animal seemed to have a bit of an eye infection, which would cause inflammation and thus explain that very high ESR value. So given that experiment stage had no effect on ESR and ESR was not associated with cortisol, it seems that ESR is an excellent indicator of overall animal health and inflammation, but a poor indicator of stress. And then looking at our glucose, ketone, and lactate results, it seems that handling has a mild impact on glucose metabolism, but doesn't seem to have much of any impact on lipolysis, which is the breakdown of fat. And finally, thinking back on my study, I did regret not measuring glucose, lactate, or ketones at capture and recapture, as I think it would have been really valuable to have those baseline metabolite levels to compare my later procedure and recovery levels to. Now we'll discuss a bit of my heart rate conclusions. So this study was the first to use heart rate as a stress indicator in elephant seals. When we looked at the general heart rate patterns, heart rate was generally low and stable during anesthesia, and then post-anesthesia, it gradually increased and stabilized within a few hours. Seals displayed highly individualized heart rate responses to transport and short-term stimuli. And I found that oftentimes the aggregated data, so when we look at just simply the means, that actually masked some of the tachycardia that individual seals were experiencing. So for example, in the short-term stimulus analysis, I found that mean heart rate significantly increased from baseline to events for only the release stimulus. However, for every other stimulus that I looked at, there were individual seals that exhibited tachycardia, and some seals exhibited tachycardia during transport to the release site. Thus, I think when researchers are thinking about the impacts of their handling activities on study animals, it's really important to look at individual responses in addition to that aggregated data or the mean values. And finally, I really think that heart rate is a valuable but underutilized stress indicator for wildlife and for marine mammals in particular, and so that we really just need more research to kind of unlock the potential of heart rate as a stress indicator. And with that, we've now reached the final discussion slide in my talk, and so I'd just like to leave you with 3 closing thoughts. So I found that intensive research handling activities tended to elicit mild short-term physiological stress responses in juvenile northern elephant seals, but most individuals recovered rapidly. And 95% of our study animals were resighted in the wild up to 2 years later, which suggests participation in this translocation study had no major long-term deleterious effects on study animals. And finally, looking at all of this data together, while I believe that researchers should always take care to minimize unnecessary handling and reduce stress loads whenever possible, it does seem that northern elephant seals are a robust species for ecological and physiological research. All right, and with that said, we have reached the acknowledgements. So I've got a lot of people to thank, so please bear with me as we make our way through this very hefty list. So first and foremost, my biggest thanks to my amazing thesis committee led by my advisor, the brilliant Dr. Gita McDonald. Gita, I don't even know where to begin. It's been such a pleasure to be in your lab and to be your student. Thank you for taking me onto this project. Thank you for teaching me about stats and coding and physiology and elephant seals and all of the things. It's just been such a blast. And I'm so happy that I joined your lab group sight unseen and moved to California. Alongside Gita, my other 2 committee members were Dr. Allison Hindle and Dr. Scott Hamilton. And I just really lucked out here with a fantastic committee. All 3 of you were so kind and supportive and offered such useful and valuable feedback at all stages of this project. So I feel really lucky to have had this amazing thesis committee in my corner throughout this long project. So as you can imagine, translocating elephant seals requires a lot of people. So here's a big list of people that I want to thank. So our larger NSF-funded project was led by an amazing team of 7 principal investigators: Gita, Allison, Cass, Paul, Marcus, Holger, and Dan. And y'all are simply the best of the best. It was such a pleasure to work with you all, to learn from you. Thank you for teaching me so much. And yeah, I really had such a wonderful time on this team. And there were 2 other grad students on this project, Sean and Daphne, who just were the most amazing partners in crime on this and truly believe me when I say this project would not exist if not for the very hard work of Daphne and Sean processing my millions of blood samples. So thank you both for your help with all of the things. We also had an amazing and very large field team who worked very, very long days in this field to make the study possible. So thank you to Rachel, Logan, Patrick, Jason, Chavi, Parka, Emma, Luke, Arena, Teresa, and Luis, and the Rangers of Año Nuevo for excellent help in the field. And I wanted to give a special shout out here to Dr. Rachel Holzer, who actually took me to Año Nuevo on my very first day back in 2019, and over the years has just taught me so much about elephant seals and how to be a better field biologist. So thank you, Rachel. I also owe a huge thank you to Dr. Dan Crocker and his grad student, Allie Northey, who assisted me with my hormone assays up in Dr. Crocker's lab at Sonoma State. So thanks, Dan, couldn't have done it without you. And finally, I had two great interns, Rebecca and Logan, who helped me analyze our mountains of heart rate data that we had. So I really appreciate their help. In addition to a lot of people, this study also required a lot of money. So huge thank you to our many funding sources, especially the National Science Foundation, which funded the majority of this project, and personally paid my tuition and salary. Additional funding support was provided by CSU Coast, Moss Landing Marine Labs, San Jose State, and Myers Trust. Next up is my lab group, the MLML Vert Lab, who I love all these people so much. I really cannot imagine a better group of people to go through the highs and lows of grad school. It's been such a blast. You guys always kept me laughing, put up with my ridiculous stranding projects and stranding responses, and just gave really valuable and helpful feedback on this project. So thank you all. I will always be a member of the BART Lab. I love you guys. Thank you. Outside of my lab group, there's just an amazing community at Moss Landing in my cohort. Huge shout out to my very dear friends, Alora, Hannah, and Chernell. I can't imagine the last 3 years at Moss Landing without you. I also had two great supervisors for my student jobs during my time at Moss, Katie Lage and Kathleen Donahue, who were both so kind and supportive. And finally, the MLML admin, who just really do so much to help the students and keep us on track with our projects and the administrative components. So thank you all. And now I need to thank my family and friends outside of Moss Landing. So I have the best friends in the world. Everyone says that, but it's really true for me. So thank you to my dearest and best lifelong friends, Jenny, Aaron, Beckett, and Cassidy. I think most of you are on this call today. Hi, I love you. Thank you for everything. And to my family who just, my parents, so there's no more supportive parents in the world. And hi, Mom, I know you're on this call. Thank you for everything. You know, I wouldn't be here without you. And finally, my last thank you in this very long run of thank yous is to those elephant seals themselves. So while researching the effects of handling on stress levels, it was really never lost on me that it was 19 very real animals experiencing those physiological stress impacts. And it really was just an honor and a privilege to work with these animals. And if nothing else, I do hope that the results of my thesis project could in some way further marine mammal welfare and conservation efforts. So thank you, elephant seals. I will miss you all very much. And with that, I would like to thank you all so much for attending my thesis defense. And I would now be happy to take any questions. Thank you so much.
Great job, Lauren. I really enjoyed your presentation.
Thank you.
If anyone has any questions, please raise your hand. See lots of applauses, but I don't think they're. You did such a good job explaining it. One of my questions that I often ask people, but you sort of answered that a little bit already, is you did a good job of interpreting what you found. If you could redo some aspects of
yes.
what are some of the things you would change? Like, I know you take more blood.
Yeah, definitely more blood samples, even though we already had so many. So it feels kind of wild to wish I had more, but yeah, more blood samples. Oh gosh, I think I probably would have done I don't know. The blood samples, I really think were the big missing thing. Maybe some other parameters, maybe look at some other stress hormones, but that's just making more work for myself. So I think it would really just be to collect more blood samples to kind of look at more of those fine scale changes.
That sounds good. We'll try to think that one after transit will be hard, but
I know, right?
we should have gotten the ones on the beach though.
Ah, well.
It looks like Sean has a question for you.
Yes.
Great job, Lauren. Really,
Thank
really
you.
good. So happy for you. I don't know if I missed this. If not, I wanted to ask, did you notice any differences in heart rate with the animals that were, or any other levels of that you measured with animals that were able to recover overnight versus were just recovered during the day after anesthesia?
Yeah, so that's a really good question. I might pull up my slide here. So yeah, we basically broke down the recovery stage. So for the blood samples, there was just early recovery and late recovery, but we chose to split it up a little bit more for the heart rate data. So we did kind of want to tease apart any differences during that mid-recovery period. So you can see our sample size for the mid was 11, which was every animal we collected heart rate data from. And the late recovery was only 5. And so that was only for those late recovery animals. And there really was no significant difference looking at the big changes. And I didn't look at that on the shorter term stimuli, but I would guess it just seemed like at a certain point, they seemed to fall asleep and just be hanging out in their cages. And so it didn't seem to matter necessarily that much, at least in terms of their heart rate responses, whether it was for a shorter recovery the same day or the late recovery for the animals held overnight.
All right. Looks like you have another question from Scott.
Hey, Lauren, nice job. Great talk.
Thank you.
I forget if I asked this or not when you were you're writing up your thesis, so I'll ask it again. I mean, you had I guess 4 males and 7 females. Were there any difference between those two sexes in any of these hormone or blood responses or anything like that?
Yeah, that's a great question. So we didn't mean to have so many more females than males in our study. That was really just accidental. It's really hard at this age class. You can't really sex them apart unless they're lying upside down on the beach. So it just so happened that we had more females, but I did look at the effect of a bunch of different parameters, including sex, on all of these changes, and there were no significant differences between sexes.
Do you imagine that there, I guess, as they got older and maybe hit maturity, do you imagine that there would then start to be some larger differences?
Yeah, I definitely think so. There's a lot of research going on looking at different age classes, and in addition to that, different the time of year. So whether they're molting or breeding, I think there's a lot of variables that could impact that, but they definitely do see sex-specific stress hormone differences. So, but it seems like at this age, that's really not a factor yet, at least in my study.
Thanks.
Yeah, thank you.
So it looks like you got a question in the chat. It's a little long, so I don't know if it's easier for you to read rather than me. So
do you want me to read it? Should I read it?
Here, I can read it, but just let me know if it's so. Basically, it's from Mike, and he's saying the internet connection is unreliable, so he's typing his questions. So can you speak a little bit about the possible differences in baseline cortisol and aldosterone of the seals compared to similarly sized terrestrial mammals? And what's your current hypothesis for what makes it about marine mammal, or what about marine mammal that makes aldosterone so much better of an indicator for acute stress?
Okay, yes, so I think I'll pull up my aldosterone slide here. So yeah, aldosterone is really interesting in terms of a stress hormone. So for most other terrestrial mammals, aldosterone is not considered a stress hormone. Its primary function is considered water regulation. So it's really interesting that in marine mammals and elephant seals in particular, we're seeing that aldosterone is a component of the stress response. So I don't know the hypothesis. I feel like I've been digging into the literature, and it seems like there isn't, I don't know, everyone's just like, oh, it's a component of the response, but we need to kind of tease out more of why it is. So I don't necessarily have an answer to kind of what function that's playing, but I think it's a really interesting line for future research. And I'm very, very interested. I'd like to know more about how it's impacting other marine mammal species in response to other stressors over longer time periods. So that's the aldosterone question. And then it seems like there's another part of your question that is about the large variation in beat to beat heart rate during transit and what you think might be going on there. Yes, so for that one, I'll pull up this heart rate plot again. So yes, large variation in beat to beat, or maybe that would be mean heart rate. So yeah, I think that this kind of just speaks to the fact when we're looking here at mean heart rate, we've got that much larger purple box for transport. I think that it kind of breaks down almost getting at different personality types of our animals. So for some animals, they seem to be quite calm and during transport, and the act of being in the truck didn't really have much of an impact on their heart rate at all. And many animals even experienced kind of their regular sleep apneas that they had been displaying previously. Whereas for other animals, they definitely seem to be a bit more stressed out during that transport process. So whether their heart rate was either higher consistently or much more variable, or they just stopped displaying all of those kind of variabilities together. So yeah, it just seemed like they still kind of fell into different categories, whether they had no change relative to recovery or they seemed to be quite stressed. So that was kind of where I was getting at. I think it's important to kind of tease apart differences where if you just look at, say, the mean here, those lines are very similar, but then certain animals were definitely more stressed out during that transport process. So yeah, I wish we had a bit more data there, maybe another 10 animals. That's something I would do, Gita. Some more animals, a bigger sample size, which, you know, is so easily said and then requires so much more work on our part. But yeah, more animals just to see if there were more clear groupings of those heart rate responses during transport.
All right, well, I don't see any more questions and we are a little after 1. So great job, Lauren. We'll have you
Thank
stick
you
around with your community while everyone else can head out. We're sorry we can't go have a party with you.